Dr. Preller, thank you for joining me.
Well, thank you so much for the invitation. I'm really glad to be here.
Yeah, we're glad to have you. Where are you sitting in today?
I am sitting in my home office in Zurich right now.
What time is it there?
It's 6 p.m., so pretty late on a Friday evening.
Okay, well, thank you very much for taking the time on a Friday. Can you start off by just telling everyone what you do and what your scientific background is?
Yeah, so I'm a neuro and clinical psychologist by training, and after graduating from college, basically, I joined a research department at the University of Zurich, where I did a lot of research on addiction. So, looking at the long-term consequences of drug use. And after that, I switched gears a little bit to investigate the acute and long-term effects of psychedelics in healthy participants to gain a more mechanistic understanding of what exactly these substances do, as well as in clinical populations. I spent some time at UCL in London as well and at Yale University, and am now leading a small research lab at the University of Zurich where we're investigating the effects of psychedelic substances.
And are there any particular psychedelic substances that you guys focus on?
Most of our work has been done with psilocybin and LSD at the moment.
hmm And so these are classical psychedelics that people often talk about in the context of serotonin. So I thought maybe we could start out by just discussing serotonin a little bit and get people up to speed on the basic biology there. So broadly speaking, what is serotonin and what role does it play in the regulation of things like emotion, plasticity, and social behavior?
Yeah, that's a really good question. And it won't be very easy to answer, to be honest, because serotonin plays such a big role in some people say in everything and nothing at the same time. But in general, serotonin itself is a neurotransmitter that is in everyone's brain. It's not only in the brain, but obviously in the context of psychedelics, we're mainly looking at the brain. So, in the brain, it regulates a lot of functions, and they range from appetite and sleeping to other functions like emotional processing, etc. So, it's really involved in a lot of things, and it's a pretty complicated system because the effects of serotonin itself depend on which receptor they activate in the brain. So basically serotonin is just the transmitter, but it has to activate certain things, which are the receptors docking stations, so that they will have an effect on emotion processing, for example. And there are several subtypes of this receptor, and depending on where exactly the serotonin acts in the brain and on what receptor, the effects may be different.
And so where can you talk a little bit about the receptors and serotonin in terms of distribution in the brain? Is this a transmitter that's being used everywhere or in particular places?
Yeah, it's pretty much everywhere in the brain. So obviously, in some places, the density of certain receptors is a bit higher than in others, but it's not like it's some kind of localized system. So really, serotonin acts all over the brain.
And people often talk about the serotonin 2A receptor when discussing psychedelics. So why is that? What is the 2A receptor specifically? Is that one localized at all? And what do we know about that receptor?
Yeah. So, well, first of all, when we're talking about psychedelics, usually they are substances which are not necessarily super specific for the serotonin receptor. So there are other receptors involved as well, but it's most of them are rather specific to the serotonin system, except for maybe LSD, which has a little bit of affinity to the dopamine system as well. But and here comes the important part. When we block the serotonin 2A receptor system by, for example, giving people another drug first that blocks these types of receptors, and then add LSD or psilocybin, the participants basically don't have any effects anymore. And that tells us that the effects of psychedelics are highly dependent specifically on the serotonin 2A receptor. And with that receptor as well, it is, you can find it basically all over the brain. So it's not that, we can point to one brain region and be like, yeah, this is where psychedelics act, right? It's really all over the brain. But again, in some areas with a little bit higher density than in others.
I see. So the 2A receptor is really important for the action of classical psychedelics because if you give people another drug that blocks that receptor and then you give them something like LSD, pretty much nothing happens.
Exactly. So we've done exactly those kinds of experiments. And I mean, if you're familiar with what LSD, for example, does, it's a highly altered state of consciousness, right? It's very different from your usual reality. But if you administer this other substance first, which blocks these receptors, then basically our participants couldn't distinguish between a placebo and, this combination of this other substance plus LSD, even though, they had ingested a medium dose of LSD, but there's really nothing happens. They don't feel any alterations at all. And I think that is pretty remarkable given that, usually you have this really highly altered state of consciousness. So we need this specific receptor for the effects of psychedelics to take place.
And when you and your colleagues are administering LSD or psilocybin, what are the participants doing? Where are they?
So all of our studies are conducted at the Psychiatric University Hospital in Zurich. So it is obviously a hospital environment, a medical environment, but we have decorated the rooms in a way that is more a living room-like setting. So it's not, the usual hospital atmosphere that you encounter when you visit someone in a hospital. So we've really tried to make it as pleasant as possible. But of course, when we are trying to find out what happens in the brain of our participants while they are on the substance, then of course we need to do, a little bit more technical measurements. And one of the things we do is, for example, fMRI. So we put people in a brain scanner and look at what exactly is happening in the brain while or after they have been administered LSD or psilocybin. So obviously that is not necessarily a very living room like setting, being in a brain scanner, but people tend to tolerate this really, really well. So when we started doing these types of experiments, we were worried in the beginning, if people, would feel comfortable in the scanner environment, but people usually really do. And well, after we've done, the measurements that we are interested in, then we take them back to the living room setting. That is, of course, a little bit different when we're talking about clinical studies, where it's really more about the therapeutic aspect instead of the mechanistic one. There, most of the measurements really take place in this living room-like setting, so we're not exposing them to any kind of EEG or fMRI experiments usually while they are on the substance.
I see. One more thing I wanted to discuss about serotonin is I think the typical person who hears serotonin, if they know anything about it, they probably associate it with mood and emotion because of SSRIs and how those are prescribed for antidepressant. So what exactly are SSRIs and how do they affect serotonin differently from something like LSD or psilocybin?
Yeah. So what SSRIs do is they inhibit the reuptake of the serotonin that is naturally occurring in your brain. So basically, serotonin is floating around between two neurons that need to communicate with each other. And once they have been activating the receptors at the other side of or at the next neuron, what happens is that the serotonin usually gets reuptaked in the first neuron where it gets broken down and then reassembled at one point. And what SSRIs do is they basically increase the level of serotonin, of the naturally occurring serotonin in your brain by making it stay longer in what we call the synaptic cleft, so between these two neurons, so they can activate the next neuron for a longer time period. And the difference to psychedelics is that here we're not working with the serotonin that is occurring in your brain, but the psilocybin and LSD activate the receptor directly, first of all. So basically they do the job that serotonin is usually doing. And the second difference is that, of course, the serotonin you have in your brain that is just there, it will stimulate all types of serotonin receptors. We've talked about this before already. There are a lot of subtypes of these receptors. So it will just activate the whole serotonin system. However, as seen in these previous experiments that we already talked about, is that psychedelics are really mainly focusing on this one specific receptor subtypes and not so much on the other.
Gotcha. So an SSRI will essentially increase the total volume of serotonin present in the space between neurons. And that serotonin will then go activate all of the different types of serotonin receptors everywhere that it can. Whereas a psychedelic like LSD will act almost in place of serotonin, but in a much more biased way at a particular receptor.
Exactly.
So SSRIs are so common. I don't actually know how common they are. I just know that they are very common. Is there any interaction that an SSRI would have with a psychedelic, say? So if someone was taking prescription SSRIs and then, in a social setting or otherwise, they took something like LSD or psilocybin, what would the potential interaction or effect be compared to someone who is not taking an SSRI?
That's a really good question. And I cannot at this point in time give you a really scientific answer to that. And one of the reasons why these experiments have not necessarily been performed yet is because people are worried that if you have too much serotonin, so SSRIs plus a psychedelic could lead to basically, too much serotonin activation. And this can cause what is called the serotonin syndrome, which is really not particularly a nice state to be in, and could be dangerous. So that's why that's why these experiments have not necessarily been done quite yet. However, if we look at anecdotal reports of people who are on SSRIs and have tried a psychedelic, what they usually report is quite the opposite, actually, that they have less effects than participants or people who don't take SSRIs. And the reason for that could be that by taking SSRIs chronically, so over a longer period of time or every day, right, you change your serotonin system. So your serotonin system, the receptors are adaptive. So if you increase the level of serotonin, you might have a reaction that the receptors themselves internalize, so you have fewer receptors, which could also be the case why if you don't have that many receptors anymore, the psychedelic itself has less to stimulate. That's why you might not feel the effects that strongly. But again, this still needs to be tested in scientific studies.
Yeah, it's really interesting to think about the adaptive or plastic capabilities of neurons. So with that in mind, what would happen? I'm guessing you don't give patients or participants LSD 2 days in a row. And can you comment on why that is?
So the reason for that is, well, I mean, so far there has not necessarily been a need to do that. And also people psychedelics are a substance with very little abuse potential, and there's obviously a reason for that, because very few people would enjoy 2 days of tripping necessarily in a row, right? I mean, it's quite a ride, so it's really not something that people, if you've taken a psychedelic one day, you hardly want to do that exact same thing the next day. But there's also a biological reason for that, because this overstimulation of the serotonin system leads to this what I said before, like internalization of the serotonin receptors. So you will have a much or at least not as much of a strong effect if you take LSD or psilocybin for that matter the next day. So you would just not feel the drug either at all, or at least a lot less. And you need at least a few days, like a week or maybe 10 days, for the system to get back in its normal state, so you can actually feel the effects again. And that is also some kind of, on a biological level, that is another mechanism that prevents psychedelics from being, abused on a daily basis, for example.
Mm-hmm. Yeah, that's interesting, and that's a normal response, right? This receptor. So you take a drug, it could be a psychedelic, or it could be just some other prescription drug, say, and if it's stimulating a particular receptor a lot, that receptor will tend to go down. It'll be less frequent, but then it comes back in the absence of the drug. Is that accurate and normal?
That is in general accurate and normal. I mean, it depends a little bit on which receptor system you're stimulating, right? So sometimes that happens a bit faster, sometimes that happens a bit slower. In the case of psychedelics and the serotonin 2A receptor, that happens pretty fast, which is also the reason why you don't, there's no binge consumption of,
Mm-hmm.
of these drugs usually. Whereas with other drugs, the recovery is often much faster, which is also why you can basically take them more or less every day. But in the long run, you will still need to increase the dose to get the same effects you had the first time you used the substance.
One of the other things that I noticed in your CV in terms of what you've looked at is you've looked at other drugs, including MDMA. You've looked at things like empathy and social cognition. So I thought maybe we could have a short discussion about those things and then maybe compare and contrast some different classes of drugs, something like MDMA, something like LSD, and then something like cocaine or a stimulant, say. So to start, what is empathy? How do you define it? And what are some of the important parts of the brain or brain networks that we know are important for empathy?
Yeah. So we usually tend to distinguish between two different kinds of empathy, the cognitive and an emotional part. So the emotional part of empathy is really feeling with the other person. So you see that someone is suffering and that makes you suffer too. Or same obviously with happiness, right? So you see someone is being happy, which usually makes you happy too. So that is the emotional part. And the cognitive part, or also called theory of mind sometimes, is that you understand what exactly it is that the other person is feeling. So I see someone is smiling, which means I know that he or she is happy. That doesn't necessarily mean that I have to feel happy too, but I know what this other person is feeling. So these are two different things we distinguish usually. They also, we can distinguish them on a neural basis as well, but they have highly overlapping networks. And in general, what we look at when we're talking about social cognition or empathy in the brain is usually the so-called some people really call it the social brain, and this includes a lot of the midline structures, so the structures that we see basically here in between the two hemispheres, or in the middle of these two hemispheres, as well as some more lateral parts like the temporal parietal junction, for example. So these are like over here. But these are like the key areas we look at when we're talking about empathy and social cognition in general.
And is there any interaction between serotonin 2A receptors and those parts of the brain, or these social capabilities?
Absolutely. So we've run quite a few tests with psychedelics and various social cognitive measures. One, empathy, for example. So what we've seen there is that psilocybin acutely increases emotional empathy. So how much you feel with this other person. We didn't see any changes in cognitive empathy, though. So it's not like they can name the emotion that another person is feeling any better, but they tend to feel more with this other person. We haven't done that in the MR scanner, so we don't know where exactly this originates from in the brain. But we've done other tests where we also gathered fMRI data, so where we can also localize where this effect is coming from, basically. And one was an experiment where we looked at exclusion or rejection processing, because we also know that a lot of psychiatric patients experience a lot of rejection, right?
Mm-hmm.
And on top of that, they also react more strongly to social rejection. When we think about, for example, borderline personality disorder patients, they have a very strong reaction to social rejection. And there, what we've seen there is that psilocybin reduces this reaction towards social rejection. So people are not are less emotional about being socially rejected. They are aware of it, but it just it's not that emotionally important. And when we look at the fMRI data during this experiment, we see that we have less activation in what is called the anterior cingulate cortex, which is exactly one of those midline structures in the brain. So very much in the middle of the brain, between or at the border of the two hemispheres.
Yep, most people probably haven't heard of the cingulate cortex unless they're a neuroscientist or something. So can you talk a little bit about the cingulate cortex? Let's be very concrete about where exactly it is and what do we know about it? I know there's a lot that we could probably discuss, but what would you say are the basic facts about the cingulate cortex and what it does?
Yeah, so the cingulate cortex is the area. So if you imagine, like, you have your subcortical structures like the thalamus, etc., which are like very early structures where, for example, your sensory input is first processed in the brain. And then you have the corpus callosum, which is connecting the two hemispheres. So that is on top of that. And right above the corpus callosum, the first part of the cortex, right above that, in the middle of the brain, so where the two hemispheres meet, that is the cingulate cortex. And again, it's a pretty complicated brain structure because it's not, we cannot associate it with one single thing that is being processed there. A lot of research has focused on pain processing in general there. Mm-hmm. There's also a lot of research that has associated the anterior cingulate cortex with conflict monitoring. So if you're seeing something that is or processing something that is in conflict with your expectations, for example. But also, and again, that is important for the experiment that we conducted for social rejection, which some people have also called social pain processing. Which kind of led to this idea that because it's processed in overlapping areas, that social rejection is basically the feeling, a painful social feeling, and therefore it kind of makes sense that it's processed in similar brain areas. And we know that if we do the same experiments that we conducted with psilocybin, if we do the same thing with, for example, borderline personality disorder patients, that we get an increased reaction in this anterior cingulate cortex in these patients.
Yeah, it sort of makes sense intuitively, I think, to most people who've studied or tried psychedelics. People often describe the experience as almost childlike in its qualities. And if you think about what a child is, they're a very emotionally reactive person. So they're very sensitive and reactive, both on the positive and the negative side. Yeah. So that actually makes sense to me. So you don't necessarily think about psychedelics as increasing empathy or emotional processing first. I think most people probably think of them first for the sensory effects they have. But something like MDMA, that's really, people call it an empathogen. The empathy and the prosocial behavior effects that it has are really the primary thing that are associated with MDMA. So how does MDMA work in comparison to an SSRI and in comparison to something like LSD?
Yeah. So MDMA is, again, a very difficult drug because it does a lot of things. In a way, it's a little bit closer to an SSRI than a psychedelics or classical psychedelics, because it's not necessarily stimulating the receptors that much itself, but it's rather releasing serotonin. So the serotonin you again have in your brain already gets released into the synaptic cleft, so the space between the neurons, and then can stimulate the neurons that are on the other side of the synaptic cleft. However, it is not only doing that to serotonin, but it's also doing that for dopamine and for norepinephrine, for example. So it's a very complicated drug pharmacologically, but it's more of a releaser than stimulating the receptors themselves. It does that a little bit too, but much less than a classical psychedelic. So again, a pretty different mechanism of action here. And you're perfectly right. So the social aspects are definitely the ones that people look at when we're talking about MDMA, because they are just so prominent, right? But we have done, not necessarily comparison studies, but I've written up a review paper where we compared the studies that have been conducted with MDMA on the social aspects, and the studies that have been conducted with psilocybin and LSD. And what we see there when we compare these results is that the social, or the increases in empathy and the changes in other aspects of social processing are actually pretty similar in MDMA and in classical psychedelics. So my interpretation here is that, well, maybe, we just don't talk about the social effects of psychedelics that much, because, when people take them, the first thing they notice is obviously all these alterations in, their visual system and the sensory system that, somewhere underneath the whole social changes and social perception happen, but they're just not as obvious, because, maybe they're just because all this other thing is going on as well. And that is also a lot easier to describe, for example. But yeah, when we look at the data, the alterations in social processing are to a degree pretty similar to the ones of MDMA.
Yeah, that's interesting. I think the flip side of that too is with MDMA, people focus so much on the social and empathic behavioral effects that they sometimes overlook the sensory effects. There definitely are sensory effects, I would say. What do we know, if anything, about that scientifically?
Not so much to my knowledge right now, because usually people have, alterations in sensory experiences, mostly at pretty high doses of MDMA. And obviously we need to make sure in the experience that we are conducting that, that we keep our participants safe. So we tend to administer, doses which are like in a moderate range, right? There is a bit of research on MDMA and social touch. So how that you perceive, the touch of another person in a different, usually more pleasant way. But I'm not aware of much research going on like visual effects of MDMA, for example, because they're just not as reliably induced, at least in low to moderate doses with MDMA compared to, for example, psilocybin.
Gotcha. So something like LSD or psilocybin is directly stimulating certain serotonin receptors in the brain. That's causing all of the psychedelic effects. MDMA is not doing that. It's really dumping serotonin and other transmitters into the synaptic space more. So you've sort of got more of these neurotransmitters in the brain doing stuff. And so in that sense, it's sort of like an SSRI. An SSRI and MDMA would put more serotonin into the synaptic space. So what's the interaction or the potential danger of having an SSRI in your system when you take MDMA?
Yeah, that's a good question, and can basically only give you the same answer I gave you before. That right now, at least, we just don't know. I mean, there's always, the problem that with the serotonin syndrome, right? So too much serotonin really is not good for you. So it's probably not something that people should try, even more so with MDMA. It's just probably not a good idea to kind of overload your serotonin system that much. But we just don't know at this moment, right? There will be, I'm pretty sure there are some studies going on or will be going on in the near future where we will have scientific data on that and where people are doing these experiments in a safe setting.
I see. So when we think about the perceptual or sensory effects of psychedelics in particular, they're very striking. So there's very strong sensory effects. People, literally see stuff that isn't there, or it's moving. So can you talk a little bit about the brain networks that underlie perception and how would you define perception as a neuroscientist, and how would you maybe distinguish it from simpler sensory processing?
Yeah, so perception for me, and I'm not entirely sure if that is, a common definition of that, or if everyone would share this definition. But in general, we don't usually experience just, your visual input or just your auditory input, but usually all these inputs that you receive from your environment, as well as the ones you receive from your own body, get integrated in one coherent perception. And I think there is where the difference is, right? You can have a visual stimulation and a visual processing, but eventually you have one pretty broad picture of yourself and the world around you, which is put together from different senses, right? And with psychedelics, obviously something happens there, and there are multiple things that happen across the whole brain, not only in our sensory brain regions, but even before we get to the cortex, there is the thalamus, which I mentioned before briefly. And the thalamus is usually responsible to filter important from non-important information in the world. So the world as it is presented to us is not necessarily the reality, because in reality, obviously, there are no colors, right? They're just different wavelengths. But not only that, we're largely ignoring a lot of stimuli. And that is good, because this is the way we can function in this highly complex world. Mm-hmm. Now, under the influence of a psychedelic, what we've seen there is that the thalamus seems to be losing its ability to really filter this information that we're receiving. And again, I'm talking not only about our environment, but also about the information that we receive from our own body. So this seems to lead to more information being sent to our sensory brain region. So from the thalamus, this information gets to the primary sensory brain regions, and we have different ones for all of our senses. For our visual system, for example, this is in the occipital cortex, which is in the back of our brain. For our auditory system, this is on the sides of our brain. And so we have more connectivity, more information flow to these sensory cortical brain areas. But of course, as I said before, you need to integrate all these things into, a coherent perception. Mm-hmm. And we see that these sensory brain regions, they're highly connected under the influence of a psychedelic to the rest of the brain. However, the brain areas which are supposed to make sense of the percepts that we have, to integrate all this information that we are receiving from about the current state of the world, the current state about ourselves, as well as integrating that these percepts with what we know about the world. These brain areas are loosely connected to the rest of the brain. So our interpretation of that is that we have a state of heightened sensory perception, but the integration of these sensory perceptions is different. So basically, we bring together this information differently than we usually would. And that could explain, on the one hand, why we experience things like, these illusions that you've been describing, but also why people, for example, say that, psychedelics allow them to break out of, their usual thinking patterns, why they allow them to see themselves in a new way, or see the world in a new light. because the way we put information together seems to be different in a psychedelic state.
This reminds me of Aldous Huxley, who famously speculated about how the brain worked basically in the context of psychedelics. He described the brain as perhaps acting as a reducing valve, basically a giant filter. It's actually filtering out information mostly before it actually comes into our conscious awareness. And it sounds like you're saying that something like that is actually happening. So on a psychedelic, there's at least two things happening, if I heard you correctly. One is relatively early parts of the information processing stream, like in the thalamus, are actually being relaxed. So you're filtering out less. There's actually more sensory information coming in and getting to the cortex. And then after that, the cortex itself, the association areas and the higher order areas are behaving differently. They're stitching that stuff together differently. We can just say that. So there really does seem to be a filtering mechanism that is reduced when you're on a classical psychedelic.
Yeah. So that is at least what the data tell us so far, right? I mean, it's obviously hard to test this in humans directly. So we have to, find ways to work around that. We cannot necessarily just stick electrodes in the human's brain usually and just watch the information flow. So we're working with other techniques, like, for example, connectivity measures in the brain. And there we've seen exactly that. So we have a stronger connectivity between the thalamus and certain parts of the cortex. There are other paradigms that we use as well, where we can test how information is processed. And basically all of these different measures that we have applied during a psychedelic state point towards a reduced filtering mechanism in the brain.
And then I would imagine, and anyone who's tried these things will know firsthand that the dose really matters. So a very high dose versus a medium dose versus a low dose are going to have extremely different effects in many cases. Right now, something that's very trendy, you can, people are talking about this in magazines in the grocery store, microdosing LSD. And, it's really marketed. It's really being marketed literally as almost a kind of stress ball or supplement, that you would take as part of your normal healthy living, because it's going to make you supposedly more creative, a little bit more functional or whatever. So do we actually know anything about the effects of microdosing something like LSD? And are there any potential risks in doing something like that, even at a very tiny dose, day after day after day?
Yeah. So unfortunately, we know very little. So there are various question marks around that. But let's look at the safety issue first. And again, there are many unknowns, so I won't be able to answer that question really. But we also know that LSD is stimulating the serotonin 2B receptor, and there are a lot of those receptors involved in your cardiovascular system. And we know that stimulating this receptor chronically, so over a long period of time, might have negative effects on your cardiovascular system. Now, obviously, we don't know if a microdose of LSD will be enough to induce any kind of adverse effects there. But it is an open question, so we have no scientific data on the safety of a chronic application of psychedelics. So this is just something to keep in mind. I'm not saying that it is necessarily dangerous, but it could theoretically be. So this is something we really need to keep in mind when we're talking about daily administration of LSD, for example. I hope that there will be some safety data in the future, but right now we don't have them. Coming back to the potential mechanisms or beneficial effects of a low dose of LSD. Again, we have very little data on that yet. And most of the studies that have been conducted are not looking at chronic administration, but they are just administering a low dose once and then look at the acute effects. And compared to a placebo, we see relatively little effects, to be honest. So some studies have tried to do some like creativity tests and things like that, or concentration and things that people describe anecdotally.
Mm-hmm.
And we haven't been able to find that effect yet.
I see. It's probably very difficult to detect something like that anyway.
It is. I mean, it's a bit harder, or it's a bit easier to test things like, you know, attention span or concentration and like these more cognitive measures, whereas obviously testing creativity is a much harder task to do in the lab, right? But yeah, in all these tests, we haven't seen a beneficial effect of a single dose of LSD, or single low dose of LSD. Now, that being said, when we looked at the brains of participants under the influence of a low dose, we have seen changes in the connectivity between the prefrontal cortex and the amygdala. And the amygdala is basically, to put it very simply, is like the emotional center of the brain. And what we've seen there is that a low dose of LSD seems to change the interaction with the prefrontal cortex, which is kind of controlling this emotional response. And what that might mean is that someone who has some kind of issues with regulating emotion, for example, because they are in a slightly depressed state, this could theoretically be helpful to normalize this change in emotion regulation. And obviously that probably, you know, getting rid of, you know, this you know, maybe not pathological, but kind of, you know, depressed state will obviously also make you, you know, more creative, and might also make you you know, makes it easier to focus on your work, etc. So there are mechanistic hints that a microdose of LSD might be helpful in people who are maybe not, you know, not performing at, you know, the level that they theoretically could, because of some kind of underlying problem like mild depression or something like that. But there is not necessarily any evidence for now that, you know, someone who's already performing at an optimal level will gain a lot from a small dose of LSD. Again, that being said, there's way too little research at this moment to be able to, you know, answer this question really.
Mm-hmm. So in your opinion, like right now, a very hot topic in the research world and also in the private sector is the potential therapeutic application of things like LSD or psilocybin. At a very high level, are you excited or optimistic that there will be some major positive outcomes there, or do you think maybe things are being exaggerated somewhat?
Well, so I do think that, you know, there's definitely a potential. So the studies we know and which are published right now definitely show that, especially in addiction as well as in depression, people are getting better. And that, you know, and that is super interesting. That is super promising. But the studies that have been conducted so far are often not necessarily well controlled, and they just included a few patients. So to be able to, you know, eventually determine whether we can use these substances as medication, there are a lot more questions that we need to answer. And first of all, we need to conduct well-controlled trials. We need to conduct trials with a lot more participants. We need to figure out what the best therapeutic approach is that basically comes with the therapy, because usually psychedelics in a therapeutic setting are not just, you know, you don't just give it to the participant and then leave them alone and hope that they will magically get better, but it's usually embedded in some kind of psychotherapy. So we need to figure out what the best approach is to conduct this therapy that, you know, it comes along with the psychedelic administration. And we need to figure out who actually benefits from this treatment and who not. Because as it usually is in general in psychiatry and in therapy, it's highly unlikely that this will be the magic bullet for everyone. There will be people who benefit from the experience and from the administration of the psychedelic, but there will be others who don't. And one of the major questions is to figure out, well, who are these people who respond well to this treatment, and who might not be really, or where shouldn't we administer that substance. So there are a lot of open questions that need to be answered, but the results that we have so far are very promising.
So one thing that you've mentioned so far is that the effects of classical psychedelics like LSD depend, if not entirely, then in very large part, on the 5-HT2A receptor alone. And another question that people have discussed in the literature and elsewhere is, well, to what extent can the therapeutic effects of psychedelics be dissociated from the psychedelic effects? Because on the one hand, some of these results with LSD and psilocybin are very striking. On the other hand, to put it one way, it's very inconvenient that a doctor has to administer something and stay with the patient in the room for 6 or 8 or 10 hours. So it would certainly be nice if doctors could prescribe something and it doesn't last that long, it doesn't have hallucinations or very strong sensory effects that the patient might have an adverse reaction to, let's say. So, but on the other hand, you've already told us that the major psychedelic effects depend very strongly on this one receptor. So do you have any thoughts, or is there any data on how plausible it is that we can dissociate the psychedelic sensory and perceptual effects from the potential therapeutic effects?
Yeah. Right now we can't, and this comes back to another open question when we're talking about using psychedelics as medication, is that we really don't know why exactly they have these beneficial effects. And I think we really need to answer this. We're trying to do that in the clinical trials that we are currently running. So on top of just looking at whether people get better or not, we're also trying to find out why they get better. So what is it really that makes them feel less depressed, or reduces their drinking behavior after the administration of the substance? And there are so many hypotheses out there which range from changes in the serotonin to A receptor system to changes in neuroplasticity, to more biological factors like insight into dysfunctional behavior, social connectedness, etc. So for the time being, it's really hard to dissociate these things, but we're trying to find out. And once we have clarified what exactly it is that helps people get better, then, and we know, for example, that it's the neuroplastic effects, or it's the long-term changes in the receptor system, then of course, if we know that it's just that the actual experience itself doesn't have much of an impact, then we know it's the biological factors, and then we maybe can also design something that is just targeting this specific mechanism of action. But for now, we just don't know.
Mm-hmm. So we've discussed how not only that LSD and psilocybin have quite a low abuse potential, but they might actually turn out to be useful for treating addiction to other substances. So you've done some research on cocaine. That's a very different type of drug. So can you talk a little bit about cocaine? What is it? What kind of drug is it? How does it behave differently from MDMA and LSD, the things we've already talked about? And how does that inform why it has a very different abuse potential than those drugs?
And so the main difference between psychedelics and almost all other drugs of abuse is that basically all drugs that that have some kind of addiction potential act on the dopamine system. And this is the same for cocaine. It's not just the dopamine system. There is serotonin and norepinephrine and these things involved as well. but the main action is on the dopamine system. That's why you have very different effects. You're feeling, very much awake. You're feeling like cognitively very sharp, and things like that, but you don't go into necessarily an altered state of consciousness like psychedelics usually induce. But obviously, due to its action and very fast action on the dopamine system, it's much more addictive than classical psychedelics.
I see. So it has to do with the relative difference in which neurotransmitter systems the drugs are primarily acting on.
Yes.
Gotcha. So what can you go into a little bit more? What do we know? What studies have been done so far about classical psychedelics and addiction? What sorts of addiction have people done pilot studies on, and where are we with that research?
So there are 2 studies published currently, one in alcohol use disorder and one in tobacco or nicotine addiction. Both of them, again, very low numbers of participants and not in a controlled setting, so meaning no comparison condition. Mm-hmm. But both of them have shown really, really nice results. So people drink a lot less, and in the nicotine addiction study, a lot of people stopped smoking. I think it was 10 out of 12 participants or something like that, or 12 out of 15 who stopped smoking. And I mean, these are incredible numbers, right? We have no treatment that is comparable to that. But of course, the studies are their first studies. They're basically feasibility studies. Again, not necessarily well controlled. And we need these larger trials and these well-controlled trials to confirm that. But these studies are currently underway. So we and several other groups around the world are conducting these types of studies with a larger number of participants, with a placebo or an active placebo control condition, which will help to find out if we can actually replicate the findings from these early studies. And it's also being extended to not only alcohol and nicotine. So as far as I know, there is one study being conducted for treating cocaine addiction, and another one for opioid use disorder. So we'll see if these early results generalize to other forms of addiction or to addiction to other substances as well. And whether we can replicate these early results. But again, the ones we have so far are really promising.
Mm-hmm. And would it be unique in the pharmaceutical world for a drug to be able to treat different forms of drug addiction? My understanding is when you're treating addiction, there's typically maybe one treatment for nicotine, a different treatment for alcohol, and so on and so forth. Is that the case?
Yeah, that is indeed usually the case. That being said, for some forms of addiction, we don't even have really good pharmaceuticals to treat them. But in general, yes. And this again comes back to the question: what exactly is the mechanism of action, right? Mm-hmm. And for example, in alcohol use disorder or in heroin addiction, what you do is basically block the receptors where the drug usually acts, so that you either don't have an effect when you use the drug again, or that you have an adverse effect when you use the drug again. So that is obviously very specific to the drug of abuse. However, again, a big question is, why would psychedelics be able to help all these types of addiction? Which basically means that if that is true, that the mechanism of action is very different from the one which the pharmaceuticals which are currently used for the treatment of addiction are targeting, right? So if we think about something like, for example, neuroplasticity, right? That could be terribly helpful in addicted patients, because they need to kind of unlearn associations between drug stimuli and their response, or the brain's response to them. And if you can theoretically combine that with increased insight into dysfunctional behavior, we could theoretically explain the long-lasting effects and why psychedelics may act across different substance use disorders. But again, we don't quite know yet what exactly the mechanism is. But if we indeed see these positive effects across different substance use disorders, then it is probably something a lot more general than the pharmaceuticals which are currently used.
Mm-hmm. So you're working and joining us right now from Zurich. I'm curious what the drug regulation is like in Zurich compared to the United States. So one point of comparison that's sort of interesting, or one of the ironies of drug regulation in the US and how it impacts research over here is, for example, something like cocaine is actually less regulated in the United States than these other substances like LSD. So it's actually more difficult to do research on what is arguably the worst, or at least the more addictive drug. And some of the medical benefits, because some of the medical benefits of cocaine are recognized, but they're not formally recognized for something like LSD yet. So what does drug scheduling or drug regulation look like in Switzerland compared to the United States for these drugs? And can you comment on which version you think is more appropriate?
It's actually pretty similar, to be honest. So I won't be able to comment on which version is better, because it's really it's very, very similar. But there is a difference, and the difference is that research with psychedelics has never completely stopped in Switzerland, and has so the lab I was working in has conducted the first experiment with psilocybin in humans in, I think, 1996. So, and that is quite a while ago. Mm-hmm. And so therefore we obviously know how to conduct these studies. We know how to get approval for them. It's obviously it's a strongly regulated space also in Switzerland over here. But given this long experience in terms of us doing this type of research, so we know what exactly we need to do to get approval. But also on the other hand, the regulatory authorities are used to us conducting these studies, and we have been doing this for 30 years. And you can show that you are able to do that in a very safe way, obviously. Create some mutual trust in these studies, and that makes it a lot easier to conduct these studies over here, because we don't have to figure out everything new, but we have this long experience with how to conduct these studies.
I see. So the regulations are actually similar in Zurich. How does that impact the research you guys are doing? Does it take a long time to get approvals to work with these things?
Yeah, it's actually, given the experience we have with these trials, it doesn't necessarily take too long to get approval. It just makes things very expensive, and that obviously is an issue. So there is little funding for these types of studies from the usual funding bodies, which are usually governmental, where we fund academic research. So it's hard to get money to do this research, and because it is scheduled, it's Schedule 1 in the US as well as here, that makes the research just a lot more expensive. So that is actually the thing that is difficult, not necessarily getting approval.
I see. So can you talk a little bit about where your funding actually comes from? Is it coming from private sources?
Yeah, a lot is coming from private sources. Private donations, etc. There is the Hefter Research Institute, which is supporting studies with psychedelics, for example. That is also relying on private donations, of course. We are lucky to be one of the few countries in the world, if not the only one, where we have actually obtained governmental funding to do studies in humans with psychedelics, and that is really unusual. I don't think there are many countries in the world where that is possible. So we have a bit of governmental funding, but not enough to keep our research program running.
I see. So how did you actually get interested in this stuff? I mean, you do this for a living. How did you get into that position?
Yeah, so for me, I was so when I studied psychology and then moved on to doing my PhD, I was really so the questions I had were really to understand, what happens in our brain on a neurochemistry level that kind of makes us, think the way we think, makes us feel the way we feel. So what is the contribution of, specific receptors or neurotransmitter systems that makes our brain so complex, right? Um, and yeah, and obviously one way to study that is by challenging your brain, or changing your brain by drugs, right? Um, and that's what got me interested in this type of research in the first place. And that's when I started doing the research on the long-term effects of addictive substances. And I think that was very, very educating, and it was a wonderful research project. But it could not necessarily answer the questions about, what exactly in the brain happens in a causal way. Like if you tweak, this receptor system a little bit, now what does that mean, right? And yeah, during that time, I learned about the research with psychedelics, and I was like, yeah, wow, this could actually, it's a safe way to put people in an altered state of consciousness, so that you can causally investigate the contribution of the serotonin to a receptor system to very complex processes like social cognition or emotion processing. So my fascination for that was really driven by the scientific interest in the neurochemistry of the brain. And then obviously working with these substances and also thinking about their therapeutic potential is another thing that was highly fascinating for me, that we might have something at our hands that is able to help people where it has been so difficult to find treatment for them.
Hmm. And so remind me, your PhD was in neuroscience or psychology?
Neuropsychology, so both actually.
Okay, okay. And then what did you study in college? So we actually have a lot of listeners that are around that age, it seems. And I do get questions fairly often that are of the form, I'm interested in this area. I'm going to study in college and I have no idea what to study.
Yeah. So I studied psychology with a focus on neuropsychology and clinical psychology. So that was basically that is my path. And then during my PhD and later on as well, I focused maybe a little bit more on the neuroscience side of thinking things in terms of brain imaging and these things. I think there are multiple ways, if you want to conduct these types of research. There are a lot of ways of how to do that, right? So our team is very interdisciplinary. So there are MDs, of course, medical doctors, which is probably, psychiatry is probably right now the easiest path to get there, but obviously a lot of psychologists as well. There is a computer scientist, there are physicians, right? So there are multiple ways of how to enter the field, and there's no right or wrong. But what I am trying to tell people is that they should think about what exactly it is they want to do, right? In terms of what are your questions, why do you want to work with these substances? Like, do you want to work therapeutically?
Hmm.
Then maybe computer science is not necessarily the right way to do that. I mean, if you are more interested in the philosophy of these, and on all the surroundings of that, I have quite a few friends who are philosophers and are also working in this field, right? But it's really dependent on what exactly it is that fascinates you about that, and then obviously also like where are your general interests, and then then develop these interests and develop the skills you need there, and then you have the choice, right? So if you are a highly trained neuroimager, then of course you can apply these skills to psychedelics as well as to basically anything else, right? If you're a really good therapist, then you can work with psychedelics probably, but you know obviously can also help patients without psychedelics and just do whatever therapy you're trained in. So I think the really important part is to get a skill set that you are good at. And if you're comfortable with that, then you can add psychedelics on top of that. So I would never recommend someone who's a therapist and who's not, is not confident to treat a depressed patient, that they should do that with the help of psychedelics, right? So psychedelics don't make your life easier. In that case, they might even make it a lot more complicated. So get a good training in what you're interested in, and then maybe then you can add the next layer of complexity to that by adding psychedelics.
I see. So you guys mentioned that you were doing some addiction related research. Can you speak to any other studies you have that are ongoing that you're particularly excited about, or maybe just areas of investigation that you think are maybe the most important to explore next?
Yeah. So as said, we cannot run like 10 studies in parallel. So the ones which are running currently are the 2 clinical trials, psilocybin for the treatment of alcohol use disorder and psilocybin for the treatment of depression. And obviously what we're looking there is, the efficacy of these substances. So are people doing better after the treatment? But we are also trying to really understand why. So basically we have, we do fMRI scans and various tests and various questionnaires with our participants before and after treatment, trying to figure out what exactly it is that makes them better eventually. Hopefully. And I think this is, we won't be able to answer this question within 1 or 2 studies, right? Because there are so many hypotheses that we need to test why these substances could be beneficial, that we'll definitely need more work on doing that. And I think this is, the big task for the future, to find out what the clinically beneficial mechanism of action is. And there will definitely need more studies. And as I mentioned before, the other thing that I think we really should be looking at in the future is a way to test which therapeutic approach is best for treating patients. And the therapeutic approach itself might actually differ between substance use disorders and depression or other illnesses. It might even differ for cocaine versus alcohol addiction. Like all these things are things that are unknowns for the time being. And I think if we want to develop these molecules into medication, then we should target these questions sooner than later.
How common, do you know off the top of your head how common things like alcohol use disorder and depression are globally right now?
I don't know. I don't have the numbers on the top of my head, but they are very common.
Yeah, and I imagine it's actually gotten worse in 2020.
Probably. I don't think we have the numbers quite yet, but yeah, it's in these times, especially depression and substance use disorders are probably rising. Yes.
Well, Katrin, I don't want to take too much more of your time. Thank you for doing this. This is definitely the longest or the furthest away geographically I've done a podcast so far. Do you have any final thoughts you'd like to leave people with on this general area?
Well, maybe not necessarily final thoughts, but if there are any questions or anything, like please don't hesitate to get in touch with me. And yeah, I hope I can follow up if there are any questions.
How can people do that?
So the easiest way is to contact me via Twitter. So I do read the personal messages there. So that's definitely the easiest way. But also, if you just want to write an email, that's absolutely possible as well.
All right. Well, Katrin Preller, thank you for your time, and I look forward to getting this up for everyone.
Thank you very much.